Good morning to all of you present here. Today I'll explain my research article that is based on the enhancement of the lipid production in the Schizochytrium species by using the synergistic actions of the chemicals. So my name is Dr. Preeti Mehta Kakkar and I am working as a researcher and the professor in Amity University, and my research area is on the waste management to the high value co-products. So our research article basically based on the innovative method for the simultaneous improvement of the biomass lipid that contains omega-3 fatty acids as a major compound in a single reactor without compromising the biomass. So in this study, we used a different type of modulator that includes sesamol, benzyl aminopurine, and EDTA as a potential stimulant that enhanced biomass and lipid at the same time in a single reactor without compromising biomass or lipid. So this work represents the potential application of combined effect of modulators for the simultaneous enhancement of biomass production and lipid that contains DHA in a single reactor. So what was the background that we chose for this work? As we all know that the microbial lipids from the oleaginous microorganisms have gained enormous attention as they have a high content of fatty acids that is suitable for the biodiesel as well as for the PUFAs. PUFAs means polyunsaturated fatty acids that have the opportunity to increase their market share due to the growing scientific documentation on their effectiveness in improving the human health and well-being. So in this study, we chose throstochitrid that are considered as a potential candidate for the sustainable generation of high-value long-chain fatty acids, that is docosahexaenoic acid and eicosapentaenoic acid. And since microalgae exhibit two different features, means it produces high biomass in the presence of the more carbon and more nitrogen, and the low biomass and high lipid in the nutrient deplete conditions. So the stressful conditions of the nutrients for enhancing lipid accumulation may result in the cessation of the growth and overall reduction in the productivity. Therefore, the enhancement of the lipid without decreasing the biomass is a prerequisite to getting the economically viable process for the biomass production and the lipid production. So keeping in mind the importance of the study, so the aim of the study was to develop the promising fermentation strategies that can augment the high lipid production along with the high biomass production from the algal soil cells in a single step fermenter without giving any stress. So the present inventions solved the problem of low lipid productivity, uh, low lipid productivity because of the starvation, and it also reduces the cost of high capital cost and the operational cost. So this approach will not only give a cost effective strategies, but it also helps in the economics, and it also helps in the problem solving of the waste management, because this study is using the crude glycerol as a carbon source which was taken up from the biodiesel industry, which is the crude glycerol was a totally waste, and we have used that waste and convert it into the high value products. So from the study, if we can say about the novelties, the novelty is like this is the proposed strategy for the enhancement of the high value lipids along with the high biomass without any compromisation in a single step reactor. And in our knowledge, no study report so far is available on the continuous simultaneous production of the high biomass and high lipid productivity in a single step without giving any nutrient starvation using a combined addition strategy of the modulators. And this sustainable and cost effective process can remove the economic constraint of the large scale production of lipids in a series of reactors, thus reducing the cost, OPEX, and the operational cost. So in this slide, I would like to differentiate that how our invention is different from the prior art. So first is this is a continuous fermentation process where the simultaneous continuously production of the lipid and the biomass are producing without compromising of anything in the reactor. And this process is like in this we have used the unique ratios of the stimulator that has been not used before in the synergistic way in the previous study. So there are some of the results that I would like to show here. So the simultaneous enhancement of biomass and the lipid was assessed by culturing the Schizochytrium strains. So based on our previous early screening experiments, the three modulators were selected like a sesamol, EDTA, and 6-benzene adenine purine that was chosen for this work to explore the biomass, lipid, and the DHA accumulation. And according to the comparison of the chosen modulators, it was observed in figure 1A and B that adding 6-BAP and sesamol to the medium significantly increased the simultaneous production of the biomass, lipids, and supplementing with the EDTA did not increase the biomass or DHA, but it enhanced the lipid. So these results revealed that the benzyl adenine purine therapy caused the synthesis of the PUFAs, which in turn increase the fluidity of the cell membrane, and as a result, it may promote metabolism and process improvement by accelerating the consumption of substrate in the medium by cells. So these findings also show that the consumption of the substrate in the medium by the cells. So these findings shows the proper concentration of the sesamol influence the synthesis of lipid, DHA, and biomass by the Schizochytrium species. So if we see here, so overall considering the preliminary work, so we can observe here that the 10 mg per liter of the benzyl adenine purine, 200 mg per liter of the sesamol, and 1 gram per liter of the EDTA were more efficient in enhancing the biomass, DHA, and lipid productivity. For this reason, these concentrations were selected for the subsequent studies to find the best synergistic combination of the modulators to maximize the productivity in a continuous system. So after the comparison of different concentration of the modulators, we chose a selective concentrations and we made the synergistic combination of all the modulators, and we observed that among the different combination of the chemical modulators, the higher biomass productivity of 13 grams per liter per day, the lipid productivity of 7.5 grams per liter per day, and the DHA productivity of 3.25 grams per liter per day were observed with the supplementation of the combination of 6-benzyl adenine phosphate, sesamol of concentration 200 mg per liter. When the optimal concentration of the 6-BAP and the sesamol was added, the lipid and DHA content reached 55% of the cell dry weight and 43% of the total fatty acids, which were 1.4 and 1.56 fold higher than that of control respectively. So it is also clear that adding sesamol to the 6-BAP medium at an ideal concentration could result in a significant boost of the DHA, lipid, and biomass productivity. The capacity of the sesamol and 6-BAP to improve the permeability and the fluidity of the cell membrane and thereby enable more substrate utilization is thought to be the cause of their synergistic actions. So the results that we produced in our shake flask experiment was also visually observed under the microscope that coincide with the study, and which showed that the strain grown in the supplemented mixture of the 6-BAP and sesamol medium has the highest number of visible lipid bodies and cell size that ranging in between 15 to 22 micron compared to the control that was only 7 to 11 micron, which shows the presence of high lipid body and which explain the better lipid yield as compared to the control. These microscopic findings were in line with the analytical results that observed for the strains in supplemented medium obtained in the previous screening and scale-up experiment. The result obtained in the present study indicated that the optimized concentration of the combination of 6-benzyl adenine purine and sesamol showed a positive effect on biomass, lipid, and DHA productivity. So after optimization of the concentration and the optimization of their combinations, our next experiment was to know that at what time we should supplement the combinations of modulator in the fermenter. Since we all know that the strains like a Schizochytrium or other bacterial strains, they have went through the 3 stages like the biomass, the lipid synthesis, and the stationary phase. The combined 6-benzyl adenine purine and sesamol addition point may have a significant impact on the simultaneous production of lipid and DHA. And experiments were conducted to add these modulators in a batch culture at 0, 12, 24, 36, and 48 hours respectively, and to investigate the optimum time for the combined chemical modulators supplementation. So here we observe that in comparison to the control, adding of these modulators in the initial stage of the fermentation, that is 12 hours, resulted in the accelerated cell growth, whereas adding these modulators in the log and stationary phase, that is 24 to 48 hours of the fermentation, had little effect on the cell growth. However, the addition of these modulators in the early stage of the fermentation significantly increased the lipid yield to 15 grams per liter from 7 grams per liter, and DHA reached to 6.4 gram per liter from 2.23 grams per liter. So on the other hand, the results showed that the lipid and the DHA content gradually enhanced with the extension of fermentation time. Lipid and DHA content were basically stable until the fermentation of the 36 hour. Similar to the growth, the addition of these modulators at early point has a greater impact on lipid and DHA as well than adding it in the middle or the late stages. Thus, adding of these modulators was observed best at time of 12 hours for the further experiments. With the concentration and with the combinations of the chemical modulators. So the results in the batch fermentation described that the biomass productivity, lipid productivity enhanced by the addition of the combination of the BAP and the sesamol. Hence, the fed batch fermentation strategies was combined with the supplement of these modulators during the fermentation of 120 hours to achieve the increased biomass and high value lipids. As shown in figure as well, the fed batch processes of the cell growth and the lipid accumulation were similar, gradually increasing with the time during the pre-fermentation stage, that peaking high at 96 hours and then gradually decreasing. Notably, it was clear that the fed batch fermentation produced the high final biomass of around 57 gram per liter, lipid of around 63.9% of cell dry weight, and the yield of 45% of TFA. And these values are quite higher, around 5 times higher than those of the batch control that was run without any modulators. So after optimization in fed batch, and we got to know that the biomass enhancement was seen, lipid enhancement was seen in the fed batch fermentation. So our final aim was to develop the continuous system for the simultaneous production of the lipid and the oil by using the synergistic combination of the chemical modulators. So to evaluate the relative influence of the combination of the modulators and the substrate for the biomass production, lipid and DHA production, the continuous cultivation system was established in the current study. And the continuous fermentation was set in such a way that the biomass is growing, the lipid is growing at the same time, and we can harvest the both biomass and lipid at the same time while removing the excess permeate out of the reactor. And in order to ensure the negligible carbon source from the permeate, and still we got the high productivity, the continuous operation was made to achieve the minimal glycerol concentration feasible in the fermentation broth without giving any nutrient straight to the culture. So with the feed of glycerol around 60 gram per liter, the continuous culture of the Schizochytrium species was tested at various dilution, and we reached up to 2.5 dilution per day, and the steady state was achieved, and this where we got the biomass productivity of 48 gram per liter per day, and the lipid productivity of around 2.8 fold higher than the previous lipid productivity that was around in the fed batch culture. And we observed that the continuous provision of the combined modulator along with the other media components had an impact on the final culture performance that measures, and the present work confirms that the continuous culture is the most effective cultivation method that allowing the high biomass lipid and allow the desired secondary metabolites, that is a DHA productivity. And the advantage of this cultivation method in the production of various other bioproducts lies in the eliminating of the inhibiting effect of the excess concentration of the substrate on the growth of the microorganisms. And the analysis of the result established this study as the first to provide a strategy of the addition of the chemical modulators like benzyl adenine purine and sesamid together to promote the simultaneous biomass lipid and DHA production in the Schizochytrium species. So after optimization in the fed batch, in the continuous one, this combined table shows the difference between the productivity that we achieved in the batch, fed batch, and the continuous one, and we got the maximum biomass productivity in the continuous fermentation as compared to the fed batch and the batch. And this findings provide a significant insight for enhancing the process productivity in the Schizochytrium species through the combined modulators supplementation strategies in the commercial application. So out of all the results we achieved that will help in the reduction of the cost of the biodiesel produced from the heterotrophic microorganisms. And these strategies, since we use the effluent from the industry that can be used as a nutrient, and we use that low value product and convert it into a high value product aiming at a desired zero waste, will improve the economics of the full process and make it sustainable. So as we all know that the global omega-3 fatty acid market was valued around 4.1 USD billion in 2019, and it is projected to increase up to 8.5 billion by 2025. And therefore this process will not only provide a leverage to the cost, financial rewards to the company, but at the same time, it produces the high value co-product that is beneficial for the manpower as well. So, okay, that was the presentation basically based on the production of the omega-3 fatty acids along with the lipids and the biomass. And this slides is only showing the technical advantages. So any question regarding this slides, regarding the presentation, the platform is open.